Hello and welcome to our tutorial of how to use our human genome visualization tool. This project was designed to help biology students develop a deeper understanding of genetics, connect the foundational terms and concepts to the central dogma of molecular biology, and represent them in visualized examples with actual human genomic data. Our tool can be accessed from the URL human-genome.herokuapp.com. At the top of the page, you will see an introduction that provides some background information on the Human Genome Project and major advances in modern gene sequencing technologies. Further reading on the Human Genome Project can be found by clicking on the link. Additionally, we introduce the central dogma of molecular biology as the process by which DNA functions as the genetic code for all living things. Before we dive into the details, it is important that students understand where we are going and the motivation for studying the topic of genetics. In part one, we introduced the genome. For students who are new to the topic, we embedded a short animation video to introduce the terms genome, chromosome, gene, and DNA, with a nice animated visualization of their relationship to each other. The first interactive visualization explores the genome by comparing the relative size of each chromosome and by providing counts of chromosomes, genes, and exons across the entire genome and by chromosome. Definitions are available on hover of each of these terms. Additionally, you can see the relative size and scale of each chromosome represented by the area of the box, and information about the number of DNA base pairs, of the size of the chromosome, number of genes within that chromosome, and the number of exons within those genes, as well as a visualization of the distribution of those genes across the length of the chromosome. The next visualization dives deeper into the composition of each chromosome, providing total counts of genes, subgene components by chromosome. By clicking on a desired gene, it filters the sums below. So you can see for chromosome 2, how many CDS regions, how many exons, how many start and stop codons, total transcripts, and UTRs are included. To reset the view, click in the white space above the bar chart. The next visualization demonstrates that the majority of the genome is not actually contained within genes, and 60% of the genome consists of junk DNA, whose function is not very well understood. A breakdown of gene versus non-gene by total length is also available by chromosome. You can see that there are differences across the chromosomes as far as how much and what percentage of each chromosome is contained within genes. In part two, now that we've explored the high-level structure of the genome and the organization of it, we will dive deeper into the function and structure of genes, select a given gene to trace all the way through the central dogma of molecular biology. A short animated video helps introduce the concepts and orient students to the location of the different gene expression steps within the nucleus and cytoplasm of a cell. Below, the student may select a gene of choice to explore within all of the charts below. The default selection is gene TP53, which is one of the most closely studied genes in the world due to its central role in a wide variety of cancers. But the student may select any gene of choice by clicking on the dropdown and either scrolling or starting to type and selecting a gene that you are specifically looking for. That auto updates all of the below charts to look at that gene that was selected. For this demonstration, we will look at our favorite gene, TP53. Below, you will see some basic information about that gene, its name, the chromosome that it's located on, the DNA base position that it starts at, where it ends, and the gene length. Here you will see a visualization of where that gene is located in teal, as well as several of the genes that are nearest to it in that region of the chromosome. You can also see that we are displaying a region of chromosome 17. The next visualization zooms into that gene and displays the gene's subcomponents and their locations within the gene. Genes consist of exons and introns. Introns are the white space in between the exons. And exons are divided into coding DNA sequences and untranscribed regions. You can zoom in to look at a crowded space within that gene and double-click the white space to reset your view. Counts of subcomponents for this gene are included below. You can see the relative number of each of those three subtypes of subcomponents of the gene. In the next section, we introduce the steps involved in gene expression and include a simplified representation of transcription, splicing, translation, and protein folding, and the process that that gene in its transcript will go through once that gene is activated. Below, we show for the selected gene, that process happening to its actual transcripts. Step 1 is to transcribe the DNA into RNA. Step 2 is to splice out the introns and the untranscribed regions, turning that RNA into mRNA. And then the final step is the mRNA, which are just the remaining CDS regions, get translated from mRNA into an amino acid sequence. Down below, you can see some information about that amino acid sequence that is coded for by the selected gene. Total number of amino acids included in the sequence and the exact full sequence of amino acids with each letter representing a different amino acid. Now we have an interactive chart that summarizes the total count and relative frequency of each of those amino acids from that sequence, with the most prevalent at the top and the least prevalent at the bottom. After amino acid sequence has been created, it folds into its final 3D shape and is now a protein. So here you can see the protein name for that sequence that gene coded for, cellular tumor antigen P53, and a description of its function, which is as a tumor suppressor in many types of tumors, hence why it is studied so intensely in the oncology and cancer research spheres. And down below, we see the actual 3D visualization of the structure of that folded protein. You can click and drag to rotate the protein to look at different sides of it. You can use your scroll wheel to zoom in to get a wider view, or zoom in to get a closer view of that protein. By hovering over a certain section, you can see in the bottom right which amino acid you are looking at. By clicking it, you can get more details and visualize the side chains of the amino acid and how they interact with each other. To reset the view, click in the white space to the left. If you're looking for a specific amino acid among the sequence and want to know where on the protein it's located, the sequence on the upper right lets you click and zoom into exactly where that amino acid is. Again, click in the white space to reset the view. For more information on the sources for the data behind our visualizations and some of the tools we use to visualize the 3D protein structure, we have links below and more information on the authors. Thank you very much for your time.